we are talking about SNPs, SNPs, and those ones which are non-coding, so regulatory ones. And as you know, it's actually very surprising sometimes, but 95% or even more of those SNPs which are actually associated with diseases of various types are not coding. So they are not located in any gene. And they are not actually changing the expression, they are not changing the structure of the protein. So instead, they are located between the genes or in the promoter of the gene, enhancer of the gene, and changing their expression. They are changing expression of the genes. Although, since we know more about the coding, of the proteins, most of the attention so far is put into those SNPs which are located in the proteins, and indeed they actually damaging the structure of the proteins. They are changing the structure, and they may influence a lot on the disease. But actually, we are losing 95% of the information when we're just focusing on the coding SNPs. That's why we want to change that. We want to look at the non-coding ones, and especially with a focus today on COVID-19 susceptibility. So we use data from this very nice website where several groups started to collect genome-wide association studies and made analysis of that data to learn and to determine genetic determinants of COVID-19 susceptibility. So basically predisposition of the people to be ill or severe ill from this COVID infection. So we'll use this data today to demonstrate how we can analyze regulatory SNPs. So regulatory SNPs, as I said, they are located in the regulatory regions, basically between the genes, and they can do a lot. They can change many things in the DNA, but our focus today is on those SNPs which can change the binding sites for transcription factors. So they can change many things. For instance, they can change binding sites for microRNAs, or they can change stability of some complexes or anything. But majority of effects, as far as we know, is actually because of changes of the DNA binding properties of transcription factors. So, for instance, here we have AP1 transcription factor, and in case if a certain mutation or certain variance is observed in the DNA place where this protein is binding, then the site can either be gained or lost. For instance, there are cases, known cases of variations or mutations which are actually disease causative, and they are located not in the genes. So, for instance, this change of A to C in this position of TCF1 gene, in the promoter of that gene, is known to be associated with diabetes. And when this A is changed to C, actually the site for COOP or DR1 is lost. So, and again, another case when this mutation in this gene and this variance, in fact, leads to a new gain of a new site. So in a T, when there is a T here, there is no site, and when the T is changed to C, or the variant is C, then the site can be observed here, and this change is known to be related to this particular disease. Actually, one change of position can in the same time kill one site and create another site. So like in this case. So all these cases are either predicted or known, but basically there's a lot of them in genome. And to do the systematic search of those cases when we see the SNPs and we want to see how these SNPs are changing the potential binding sites in genome, we are using TRANSFAC database, the golden standard of the collection of predictive models for predicting binding sites for transcription factors. And currently, we have around, we actually have 7,600 different positional weight matrices for vertebrates, vertebrate factors. Just top of that list is presented here. So you see basically the name of the transcription factor family, or individual transcription factor, which is binding to the motif, which is presented here by this logo. And we have 7,600 of these positional weight matrices, which then can be used basically in the following way. So if we have a matrix like this in TRANSFAC, and as you know, this matrix was built most of the cases by collecting known sites, for instance, for one transcription factor, say E2F1, and then the matrix is calculated based on the frequency of nucleotides which were observed in the particular positions of those sites. And when the letter is high, that means that this frequency, so in this position, always say C or T was observed in the real sites for E2F1. And now, when we have this matrix prepared of some known sites, we can apply it to a new sequence, to a new promoter sequence, or a new genomic sequence, which we believe is involved in regulation of a gene. And then we can actually calculate a score, which means how well this position is actually fit to this consensus, or more exactly to this positional weight matrix. But what happens if a particular position is in this place is changed? So we see that in one case it was T, and then it's changed to G. So then immediately, using the same matrix, we can calculate a new score of that place, of that position. And then in case if we see the drop of the score, for instance, the new score after this change is much lower than the original score, we can say that this site was lost. And in other way, if this Q is becoming bigger and much bigger and bigger, higher than a particular threshold, we can say that this site is gained. So it appears in this place. And in fact, we know that this binding of transcription factors to the places in genome are not exact. So we should forget this concept of exact fit. In fact, they are binding very proximate, and it leads to the problem that actually the same place of genome can be bound by different transcription factors. We can predict many different sites on the same place. And also we know experimentally that indeed in different situations, different conditions in the cells, this place in genome can be targeted by different transcription factors. So and if we observe a mutation in that place, it can influence not just one site, but several of the binding of different sites. Moreover, this is actually observed in vivo. Here you see example of experiment which was done in a cell line and colon cancer cell line actually, where an enhancer of a MYC gene, there is one SNP, and one variant of a change of a nucleotide. And in that line, which carries this SNP, the experimentally, we observed a lot of binding for many transcription factors to that place. Actually, the whole place is rearranged. So in case of this SNP, not only the binding sites which are immediately affected by the SNP are affected, but also the region around is getting rearranged. And why is that? Because we know that transcription factors, they are not actually binding alone. They are binding in accomplices, so-called composite modules. So when the binding sites for a central transcription factor, say in the center, say this is a factor RAF and it's binding to this place. But in nearby, there are other binding sites for other factors. And only when these 3 or 4 or 5 transcription factors coming together and binding in synergistic manner to this region, then the particular change of expression is observed in that gene. So, and we come with this concept of fuzzy puzzle. Basically, since this binding is not very exact, then one mutation in place can distort the whole region, and the complex of factors can be substituted by another complex of factors, which would drastically change the expression of the gene. So, and to analyze that, we come up with an idea to apply our composite module analysis, the algorithm which searches for the composition of binding sites, not just at the place where the mutation is happening, but at the region around this place. So that's why we can not only look at the transcription factor binding sites, which is immediately affected by this change of a nucleotide, but we can also look at how the whole region is rearranged after the mutation. And here we have this algorithm, so I will not go too much into details, saying just this algorithm is actually applying genetic algorithms. It searches for the combination of transcription factor binding sites in a given region. And at the end, it gives such output where it come up is a composition of transcription factor matrices, which you see here. And then each region we are looking at is then searched by these matrices, and then the algorithm finds clusters of them. Basically, you see these gray areas where the region of genome near the binding, near this change of nucleotide, near the SNP, is actually enriched by a cluster of those transcription factor binding sites. And actually, you see that if we compare the concentration of those clusters, those binding sites, in the regions with mutation, and compare them with other SNPs not related to this disease, for instance, then we see clearly separation. So these regions around the SNPs related to a particular disease, they are saturated, they are enriched by many sites for a composition of transcription factors. And the next step after we find these binding sites, we could ask a question how these transcription factors are regulated, and we are looking for master regulators in the signal transduction pathway. I will show you a bit of that today, but still our focus mainly today on the transcription factor binding story. Just to let you know that how we actually search for master regulators. We look at them in our TRANSFAC database, a database on signal transduction reactions, and we are looking for basically searches in this network for master regulators, which can lead to activation of several transcription factors in the cell. We also check if there is a certain change in or potential change in this setup. We call it working pathways, when the master regulator, which must regulate these genes in the standard conditions, in the disease conditions, is changed by a new master regulator because of multiple feedback loops. And I will touch that a little bit in my presentation today. And we see then these disease conditions, for instance, when we have inflammation conditions in the cells, they are characterized by many feedback loops when the activated master regulator leads to the activation of the genes which encode the master regulator and which are encoding the whole components of signal transduction pathway leading to those genes. So basically those red stars are indicating the SNPs. And in the disease conditions, those SNPs can lead to the feedback loops, which may determine the higher susceptibility to a particular disease.